Okay, so thank you very much, Ugo, for the introduction, and also thank you very much for inviting me here to present how we are currently investigating acute lymphoblastic leukemia in childhood. So my title is Prevention of Childhood ALL by Early Life Immune Training, Concepts and Challenges. And so I would like to go a little bit deeper into the etiology of leukemia and how maybe the host immune response may be connected to trigger acute leukemia, and maybe this may be a starting point how we can think about prevention strategies of ALL. So, as you all know, we have a very characteristic age peak in acute lymphoblastic leukemia patients between 2 and, let's say, 8 years, which is very characteristic for children with B-cell precursor acute lymphoblastic leukemia. We do not see this in other types of leukemia. And what we know from leukemia is that we have probably like two events until the progression to leukemia finally. So we have a pre-leukemic clone, often with genetic lesions which present from birth on, which then progress at a certain time point to leukemia. And we still do not understand very well why pre-leukemic clones persist in the bone marrow and what we need that they progress to leukemia. One important aspect is, of course, the presence of pre-leukemic clones, and these pre-leukemic clones can be either characterized by somatic prenatal translocations, which are only present in the hematopoietic stem cells or precursor cells, which are, for example, ETV6 rungs or BCR-ABL1 translocations. And on the other hand, we might have patients, children, who have a germline predisposition, which is transmitted from the parents or which exists de novo in the children, and which is, of course, present in all body cells. And there are also different studies exist to say that about 5% of leukemia patients carry genetic predisposition. So when you have a pre-leukemic clone somatically, so a translocation in the hematopoietic stem cells, it is still not clear why a relatively high proportion of children carry these clones, about 5% for ETV6 rungs and 0.6% for TCF3 PBX from birth on. So this was detected in postnatal blood spots, and why only a minority of children finally progress to leukemia. And the same thing is when we have like germline predisposition, so children with mutations in like hematopoietic transcription factors mainly, so which can be in ETV6 rungs, for example, IKZF1 or PAX5. Classical is probably PAX5, which is a classical transcription factor for B-cell development. And there are numerous reports who say that there are variants in these genes transmitted within families which give a higher risk to leukemia. And here are examples. So numerous variants in genes have been described with a predisposition to acute lymphoblastic leukemia, for example, PAX5, ETV6, TP53, etc. And a characteristic pedigree is shown here on the right side for a family harboring IKZF1 variants who present with either low B cells, so kind of a CVID phenotype, and certain patients also develop acute lymphoblastic leukemia. Another scenario we were working on for a long time is germline predisposition in PAX5, and this is very rare, but also here families exist in which a heterozygous mutation in PAX5 is running, leading to reduced transcriptional activity. And what is very interesting that in these affected children and carriers, always marked here with a red plus, not all the carriers develop finally leukemia, so we often have reduced penetrance. And what is also interesting is that in the PAX5 families, the children usually develop leukemia within the classical age interval between 2 and 6 or 8 years. When they have reached adolescence or adulthood and still carry, of course, the variant, they are not anymore prone to leukemia and usually do not develop leukemia. So the age interval seems to be important in connection to the genetic predisposition that leukemia occurs or not. And so we were posing the question, is there any evidence, for example, for an infection being a trigger for B-cell acute lymphoblastic leukemia? Because we say, okay, the children develop leukemia between 2 and 6 or 8 years, or there we have the peak at least. And this is the period where children first like go to school, enter kindergarten, and they are exposed for the first time to their peer groups and many infections. And this hypothesis is not new and was proposed even at the beginning of the 20th century by Ward, and then by numerous, well, really knowledgeable people in the field, Mel Greaves and Kinlan and Smith, and everybody had a slightly different hypothesis. And there are many epidemiologic data which is supporting hypothesis or challenge of the immune system being a trigger of leukemia by space-time clusters in BCP-ALL, which have occurred in the past in the USA, for example, but also very recently also in Milan. And there was a cluster or hotspot in a school with a concomitant influenza pandemia. And therefore children developed leukemia within 4 weeks. So this is really this is really remarkable to have such a cluster, and published recently by colleagues also here from the Legend Consortium, Gianni Casaniga. On the other hand, the question is, okay, when there is data that infection exposure can trigger leukemia, see the opposite thing, for example, now during SARS-CoV-2 pandemia, where we really have social lockdown and reduced social contacts. And there are data from a SARS pandemia in Hong Kong dating back to 2003. They had a pandemia between February and June, with many patients diagnosed at that time with SARS and also many mortalities. And they had also a 2-month shutdown, complete shutdown of kindergarten, schools, and an additional 6 restricted hygiene measures. And ALL incidence in that regions went down remarkably and then caught up again within the 2 following years when the social distancing measures were left aside. And on the other hand, currently we also experience a pandemia, COVID, here in Europe, and also preliminary data were released last year by the Oslo University, who also report maybe not really strong data about a reduction in ALL during the pandemia. However, there are also like cautious remarks and notes published. This is probably currently a very short observation time, and these data have been taken in account with caution. However, also experimental data exists that infection can be a trigger, and there was a huge paper from Papamanuli and Greaves in Nature Genetic in 2014, who sequenced patients with ETV6 Runx leukemia and could show secondary lesions are close to RSS, so rotting sites. So assuming that during infection, RAC and AID are upregulated and are inducing by off-target activity secondary lesions and clonal evolution. And also Markus Mission showed in his lab in Nature Immunology in a mouse model that the ETV6 Runx pre-leukemic clones in a mouse model need LPS, so or let's say LPS, so a stimulus or mimicry of bacterial infection can upregulate RAC1 and AID, and that this is relevant that leukemia can progress. So this were the first experimental data that infection might be a trigger, and we were keeping up on that hypothesis in a SCAR ETV6 Runx mouse model, so where ETV6 Runx is expressed under the SCAR promoter. And when we keep these animals in the SPF facility, so pathogen-free, they do not develop leukemia. However, when exposed to the conventional facility, they develop leukemia, and could nicely show that there also RAC plays a role because we have inactivating mutations in KDM5C, so in histone demethylases, and if the histone are not demethylated, then RAC1 and RAC2 can bind better or stronger, and that might be also an indication for more off-target activity. And this phenomenon could be reproduced in a PAX5 model. You remember I showed you the PAX5 families, where also heterozygous PAX5 variant is running in the families with a higher risk to leukemia. And also here, we experienced a higher leukemia incidence or leukemia incidence only when the mice were housed in the conventional facility. And so in terms of the PAX5 model, the PAX5 mouse model is very similar to what we see in the patients. Namely, we see here PAX5 in the patients, we have a heterozygous PAX5 variant. And then as a somatic lesion, we see loss of the CDKN2A locus harboring the wild type PAX5 allele. So the patients retain in their clonal evolution the mutant PAX5 allele with reduced transcriptional activity, and then of course acquire secondary driver mutations. However, in the mouse model, we start from the opposite phenomenon. So we start with a heterozygosity, then the PAX5 mice acquire PAX5 point mutations, and then secondary lesions which drive leukemia. So what we can summarize from the data we have on the hypothesis of infection being a trigger, that when we have a pre-leukemic state, either by the presence of a translocation or by a germline variant, infection can be a trigger of leukemia, at least in the mouse models, with then distinct somatic lesions which we can detect in the leukemia, histone demethylases, for example, EBF1, also shown for human ETV6 ranks leukemia, and the PAX5 mice characteristic PAX5 point mutations, JAK3 and activating mutations, and CDKN2A loss of function mutations. And then when we say, come to the next step and say, okay, infection can be a trigger. So is there maybe a rationale or other options for early immune training to maybe prevent this overreaction of the immune system or aberrant reaction of the immune system to infection exposure when we have a pre-leukemic clone? And there different rationales are, or different hypotheses are ongoing. So what we think currently, of course, is that we need genetic predisposition and kind of maybe an aberrant immune response that leukemic or pre-leukemic cells can grow out to leukemia. And there are many epidemiologic data, so proxies of exposure and immunologic modifiers who may pinpoint towards early immune training being protective or being beneficial to avoid leukemia. And there's, for example, daycare attendance. There are data that early daycare attendance is decreasing the risk to leukemia. Also the birth order, so secondborns have a lower risk to develop leukemia, medication prescribed in infections, and contact with livestock. And on the other hand, the mode of delivery seems to be important. Breastfeeding seems to reduce the risk of leukemia, and also the birth weight can be an issue to modulate the risk to develop leukemia. So this is all based on epidemiologic data. And there are also current data that the host cytokine response may be different in children who have a higher risk to leukemia than others. There's one study dating back to 2011, where postnatal blood spots were analyzed. They analyzed 11 cytokines at birth in a cohort of ALL and control patients, and showed that lower IL-10 levels are associated with an increased risk. And on the other hand, a second similar study also conducted in postnatal blood spots showed that soluble IL-6 receptor, IL-8, TGF-beta, etc., are associated with a higher risk to develop leukemia. And this was paralleled in 2005 by GBAS studies where polymorphic interferon gamma alleles were identified associated with a later onset of ALL in interferon gamma low responders. And these, especially the effect of interferon gamma, was later on also validated in mouse models by the group of Fidanza et al. There is further epidemiologic evidence for beneficial training of the immune system. And so I think one can say that there are probably parental factors early before birth, which might avoid leukemia. Of course, this is reduction in chemical exposure, avoidance of infection, healthy diet, and folic acid. This is what we all know. But there are also hints that training of early immunity might be beneficial, like livestock contact, social contacts, breastfeeding, or TIBVs, which stands for trained immunity-based vaccines. And also the microbiome, also an important factor for host immune response, was investigated lately. And also here there are many epidemiologic data showed by the filled bars. So for breastfeeding, social contacts, livestock, as just said, but also experimental evidence exists that avoidance of overexposure of antibiotics, which affects the microbiome, may produce a higher risk to leukemia. Use of antibiotics and healthy diet can also have an impact on the microbiome and on the leukemia, on the risk to develop leukemia. And the microbiome was also studied more in depth, also by our group and associated groups by Arndt Borchardt and Isidro Sanchez Garcia, who tested if the microbiome has an impact on the development of acute lymphoblastic leukemia. And what they did is they took the PAX5 mouse model, where we know that the mice are developing leukemia in 25% of cases in the conventional animal facility, and they treated the mice with antibiotics to destroy, let's say, the physiologic flora, bacterial flora in the gut. And what they could see that when they destroy by antibiotic use the bacterial flora, the physiological flora, the PAX5 clones grow out to leukemia at a higher incidence than compared to the control mice. So the genetic predisposition, PAX5 heterozygosity plus treatment of excess antibiotics and destruction of the microbiome can have a higher risk to develop leukemia, at least in this mouse model. However, data from patients are still missing. And a further aspect which might become interesting in the future is the concept of TIBVs, so trained immunity-based vaccines. There are also epidemiologic data, as you see here below, that early vaccination, especially children vaccinated with BCG, might have a lower risk for leukemia development. And we went back to the German, let's say, cohort or children who developed leukemia in Germany when Germany was still divided in an eastern and a western part. And in the east of Germany, BCG was vaccinated very long, down to the 90s. However, in Western Germany, not. And we had a quite statistically significant different or lower ALL incidence in East Germany compared to West Germany. And when the wall came down over the years, the incidences of ALL in the two separated countries, let's say, leveled out. And there's a concept of trained immunity, which was recently published very, very high in different tumor models, but also in the context of infection, saying that not only the adaptive, but also the innate immune system is able to acquire memory by, for example, training early in life, by exposure to infection, by vaccination via BCG or beta-glucan, for example. And that memory of the innate immune system, which is mainly characterized by a distinct methylation state, then can affect or modulate a second event later in life, for example, exposure to infection or exposure to pre-leukemic, or not pre-leukemic, but pre-tumoral or tumoral cells. And so this is also, I'm also already coming to the summary, and I see that I'm quite fast. However, so what we currently think, and I want to pinpoint that we are only talking about this early occurrence of BCP-ALL, is that we think that we need in many patients a genetic predisposition, either a somatic clone with a translocation or a germline predisposition, which gives a higher risk, and then an aberrant host immune response that pre-leukemic clones can grow out. And I think a very interesting point is, are there hypotheses or measures how we can maybe prevent leukemia, or how we can direct the immune system that predisposed children do not have such a high risk to develop leukemia, either by TIBV, so vaccination programs, beta-glucan, or others in the future. And so I'm happy that I could introduce you to our concept, our work, and the epidemiological data which are around, pinpoint to a higher or lower risk of ALL development. And I want to thank my group, especially Franziska Hauer and Mina Morkos, who have moved with me from Dresden to Munich recently. Of course, my former host institution, together with Arndt Burkhardt, who's my mentor and with whom we have worked for a long time on this concept, infection exposure and leukemia development, Isidro Sanchez Garcia and his group from Spain, and of course many collaboration partners, and COST and all the legend members. And I thank you for your attention and listening. Many thanks.
Thank you very much, Julia. Thank you for that very interesting findings and sharings. So let's start with questions.
Can I have the first one, please? So, Julia, what are your thoughts, what to expect after the lockdown that happened worldwide and the decrease rate of regular immunization? Is there anything to expect in recent times ahead of us?
Well, I'm really careful with any like looking into the future. I think currently there, to my knowledge, there are no clear data about ALL incidence and how this has changed during the current pandemic. I think the social lockdown in the different countries, different vaccination status, of course, different social habits, let's say, are very different in the different countries, even when we restrict our analysis, for example, to the European countries. But I mean, there are the publications on the Norwegian data, like from the beginning of the pandemic. There's this comment from Kjell Schmigelo, who will also talk today, who definitely says we have to be very cautious in drawing any conclusions at this early stage. So I think from my point, no really conclusions yet. But I'm pretty convinced that when you have children with a genetic predisposition, and let's say who have maybe a mild immunodeficiency, which is maybe clinically not apparent, or mild lack in differentiation of hematopoietic or precursor cells, and then you have an excess like stimulus of the immune system by going to kindergarten, and during the winter time you're sick for like 3 or 4 months continuously, that might really increase the risk that aberrant clones might grow out to leukemia. But it's difficult to prove this experimentally or in a human setting.
Thank you.
Yes, there will be more data, I believe, after these years. So please just raise your hands and ask questions. And there's a question in the chat. Oana asking if only BCG vaccination has a protective role, or the other vaccinations in the national programs have roles as well.
Yeah, so this screenshot from the study from Mora I showed, they have investigated also other vaccinations, also measles and rubella, etc. And the data are only statistically significant for BCG vaccination. However, I mean, this is a meta-analysis also like being based partially really old data. So for example, that did not dissect the different ALL subtypes. So my talk was primarily on the children who develop leukemia between 2 and let's say 8-year-olds, so classical ALL with ETV6 rungs translocations. The younger children, like below 1 year, who are, for example, pro-B ALL with MLL rearrangements, definitely have a different type of etiology, because these are very strong oncogenes. And so the primary publications who filled into this meta-analysis, they in general look over the whole bunch of leukemia patients and not specifically on this on the age interval between 2 and 8 years. So it's difficult to say if maybe also other vaccinations might have an effect if you restrict your analysis. But yes, currently you can say we have only the data on BCG having a protective effect. And maybe this is due, I mean, BCG is in that terms special because it's a live vaccine and it's vaccinated very early. So usually within the first days or first weeks of life. And this is the difference to the other live vaccines, measles, for example, which is given at a later time point, usually when children are 1 year old.
Yes, and also Anna is asking for the same issue. How would be the latency period between the infection triggering and the development of leukemia?
Yeah, so I mean, in children, we really can't say anything about it. We know that acute leukemia, I mean, they occur very rapidly. So children who are diagnosed with leukemia might have a totally normal blood count 3 or 4 weeks before. So in the human setting, we have no idea. Of course, we have many children, when they have leukemia, they come with infection. However, this can be also like a result of just the neutropenia and the leukemia who already is present. So it's very difficult to draw any conclusions from the human setting. From the mouse models, I mean, in the Markus Müschen study with the xenograft, with the xenografted ETV6 ranks cells, it was few weeks or few months only. In our model, where the animals develop spontaneous leukemia, this was between 9 and 12 months after exposure. But these are the data from the mouse model, which cannot really be extrapolated to the human setting. So it's really difficult to prove this hypothesis in humans.
Definitely. And any further questions? Yeah, it's so Marketa asking, do you think we can expect moving the peak incidence of ALL in children after the lockdown into the higher age group? And the meaning of that children with predisposition were not able to get infection triggered during the lockdown.
I can only say maybe. I don't know. Of course, one could speculate that, okay, if you have pre-leukemic clones and now like children between, I mean, they don't go to kindergarten at the age of 1 or 2, but maybe later now with 3 or 4, because we had the lockdown now for 2 years. This can be, but I think that we really have to wait for the data and the large cohorts also.
Definitely. And yeah, Cruz.
Sorry for the hand raising. Hi, Julia. I would like to ask you, do you think we can now generalize these data? These data are from Europe, Western Europe, right? Or at least...
which data?
The data on the last study with the vaccination, for example,
yeah.
The effect of the BCG vaccine. Do you think we can generalize this? I'm thinking about, I have a second affiliation at the University of Kilimanjaro in Tanzania, where we have 2 populations, one population which has a very high incidence of leukemia and the other population not. And I'm just wondering if we could maybe think that there is also a difference in vaccination grade, or whether this really is something that we were thinking in the first place, that there is a different genetical background in general between these 2 populations.
But I mean, if this is the case in Tanzania, it would be really nice to have a look if these data exist. No? But even if you say, okay, there may be two populations with a different genetic background, the one is maybe more leukemia prone, the other one not, maybe early vaccination and training of the immune system early in life by vaccination can make a slight difference. I think so. This training of immune system really only plays a role in these, like in these classical leukemia patients, no, who all receive therapy and who all become healthy. again. It will not work, I think, in the MLL rearranged, so in the very young children, and it will also not work in the adolescents, for example, which have more like BCR-ABL, JAK-STAT pathway activation, so the really strong oncogenes.
Yeah.
But if you have data, or if you have a good collaboration there, we would be definitely interested to have a look if these data are available. Yes.
Yeah. No, just to give you an idea, MLL doesn't appear there.
Yeah.
We don't see it because there is quite still a high rate of child death also. They will not be seen by us. But on the other hand, the age group that you speak about, this age group between 5 and 8 years rather than 2 to 5.
Okay.
To 8, we see this phenomenon, that there is this difference between the Maasai population and the people more coming from the coastal regions. But
okay.
Yeah, it's very stimulating to think not only about genetic predisposition in narrow sense, but in a more broad sense. And of course, these things are actually overlapping the two and brought together, right? Yeah.
And under the Maasai, then there's probably a lot of consanguinity also. So they have a very, we assume there's a protection.
No. No, it's actually not so high in Tanzania, the consanguinity.
Yeah. So the cancer complex, and not only the predisposition, but the personal and the environmental factors. And you did also just want to correct that if the early infection exposure in the first year of life doesn't protect against pre-BCP-ALL. Is that right? And how can we explain this immunologically? So, more basic.
Yes, I mean, they're the two issues. So we think that like when the child is exposed early in life to infection, or let's say has a good microbiome diversity because it is born by vaginal delivery and not by cesarean section, or for example, it is vaccinated by with live vaccines like BCG. So this is exposure to infection, let's say early in life exposure to pathogens. Also, when you're second born and when you have like older siblings, you're exposed at the first month, like with different bacteria, viruses. So your immune system is challenged. So this is definitely should be protective because you train your immune system early in life, and then your immune system works proper when you yourself probably then go to school or kindergarten with 2, 3, 4, 5, 6 years, and the first time experience like really recurrent infection, recurrent challenge of the immune system. If your immune system is not trained very well early in life, then this later challenge might be too overwhelming, especially when you have like pre-leukemic clones or preformed hematopoietic stem cells, precursor cells, which do not, for example, progress or differentiate properly. And this might be the difference. So late exposure being rather a risk to leukemia development, early exposure to infection and training of the immune system being beneficial or being protective of leukemia.
So actually this also answers Anna's next question. It was, can we transfer all the consideration about the possible infection triggers also in the adolescent and the young adult population?
Yeah, there I would rather think so. I mean, what is so remarkable in childhood ALL that we have this age peak and that these children, I mean, 95 or almost 100% of the children become healthy. We can cure them with chemotherapy. And the older children and the young adults are more characterized, as I said, by BCR-ABL positive ALL, by JAK-STAT pathway activation. And so there, I think the genesis or the etiology is different, and these leukemia are characterized by strong oncogene activations, which might be a different pathology behind. And I think we cannot transfer these concepts to.
Yeah. So the Nick also wants to correct again if the early infection is protective for some, but a trigger for the others? Or is there any difference between the exposure to immune triggers and the infection?
I think this, can you?
Early infection is protective. Or
Can you do once again, Ugor? I didn't get this.
it asks, am I right that early infection is protective for some, but a trigger for others? Or is there a difference between exposure to immune triggers and infection?
Yes, there is. Well, I think we have not enough data to say this, but as I said, so the late exposure to infection and the trigger to leukemia, there we can currently not say it's a specific virus, it's a specific bacterium, etc. My view would be that it's just challenge of the immune system. So it must be repetitive infections, either by virus or by bacteria, who then cause this stress to the hematopoietic system. When we think of the protective things early in life, there I think it's probably more the innate immune triggers. So those factors which trigger or shape the innate immune system and give a memory to the innate immune system. And this is nicely shown, for example, for BCG, for beta-glucan. So these are the classical trained immunity or innate immunity inducers.